Hello everyone, and welcome back to our next edition of our microbiology course. This chapter is going to cover how bacteria replicate, how we measure that growth, how they grow, what types of factors can influence that rate of growth, how fast they grow, and some of the things that you need to take into account when you try to grow bacteria in a lab. So let's go ahead and get started with that. So the first thing that we need to talk about when we're talking about bacteria growing is nutrition. And you're probably familiar with nutrition just from yourself or feeding your animals or pets or things like that, that you have to give them and ourselves the right nutrition for us to grow up healthy and have our bodies and cells function as they should. And bacteria are no different. When you want them to grow in a lab or grow anywhere else, even in the environment when they would need to grow, they need the proper things for them or they're not going to be able to grow as healthy and successfully as they normally would. So when we're talking about nutrition, we're talking about the process of acquiring substances from the environment, usually chemical, to convert them into a usable form of energy that can be used for cellular activities like ATP and things like that. So that's what nutrition is. It's bringing something in from the outside, nutrients, turning those nutrients into something usable by the cell. So there are a couple of different types of nutrients. There are things that are considered essential nutrients. And these are things that you absolutely must have, bacteria included, to survive. Things like phosphorus, things like magnesium, little tiny things that your body needs, things like lipids and things like that that you have to live. Without them, you will die. So there are two categories of essential nutrients. There are macronutrients and micronutrients, also known as trace elements. And their names kind of imply what they are. There are macronutrients, which, as the name implies, are large, or things that you need in large amounts, I should say. And these are going to be things that your body needs and the cell itself needs, so a bacteria would need, to make proteins, to make cell structures, to make things inside of their cell. So they would need a lot of carbohydrates, a lot of proteins from their diets, things that they can bring in, us included, that we can bring in from the environment, from our nutrient sources, and use to rebuild our bodies. And then there are things that you need in just a little bit of amount, things like manganese, zinc, and nickel, things that work as cofactors for enzymes, things that help protein structures build correctly and things like that. So you don't need a lot of these because they last for a fairly long time. You don't have a lot of stuff that's going to require these. You have way more cell membranes than you're going to have enzymes that require nickel to function properly. So you don't need a ton of this, and in fact, too much of it can be bad for you. Bacteria included, too much of this stuff can be toxic. And in fact, there's enough dissolved minerals and things in non-filtered tap water that you get your fulfillment of micronutrients from the stuff that's dissolved in general tap water, just the metals that are dissolved in there and things. So you have to keep in mind when you want bacteria to grow, you have to provide both macronutrients and micronutrients to them in the right amount of ratios because they require both of those things to function the same that we do. So you also have to keep in mind the inorganic versus organic nutrients. Now, we've covered these organic versus inorganic molecules and what they are, the presence of carbon and hydrogen going to be bound together in our organic nutrients and our organic molecules. These are going to be the biological molecules, our carbohydrates, lipids, proteins, nucleic acids, and things like that. Versus our inorganic nutrients are going to be salts, gases, and things like that. Water is an inorganic nutrient, things that we don't make. That we require in our diet. And that's going to be an inorganic nutrient. So let's talk about first why these things are important. Why do we need nutrients? Why do we need to bring in carbohydrates and things like that? Well, we have to maintain the stuff inside of our cell. As you can see here, a cell, every single cell on the planet generally is going to have this pretty basic breakdown of contents inside of them. The percentage of stuff inside of the cell itself is going to be about 70% water, a bunch of proteins, and the rest of the cell is going to be pretty much composed of 6 elements: carbon, hydrogen, oxygen, phosphorus, sulfur, and nitrogen, moved around in some shape or form to form lipids and carbohydrates and things like that, DNA, those big biological molecules that are required for the cell. So you can see here why bringing in carbohydrates and things like that to get those carbon sources, to get those oxygens and things like that, to build them for the cell are so important. Most of our cells are going to be made of those things, so we need a lot of them brought in to maintain the cell being healthy, to maintain the cell's integrity, and repair if there's any damage to the cell, and also build new ones as the cell grows and divides. Well, when we get to bacteria themselves, the same as pretty much every other group of animals or organisms on the planet, I should say, you can break them up into two main categories, talking about where they get their source of nutrients from. And you're probably familiar with the terms heterotroph and autotrophs, or photosynthesizer, producer, things like that. Consumer, you probably heard those terms before. So when it comes to bacteria, they're the same as we are, as they're plants and animals, heterotrophs and autotrophs, organisms that produce their own food versus organisms that have to get their food from some other outside source. And in this case, we're talking about carbon. Where's their carbon source going to come from and that kind of stuff. So we can break those up as well. So when we're talking about bacteria, we have photo and chemo, and that's what the two prefixes that you can either attach to heterotroph or autotroph are. So you're going to have things that are called photoautotrophs, and they're going to use energy from light and CO2 from oxygen and things like that to bring in their energy source from carbon. Their light's going to hit them, vibrate the chloroplast around, produce photosynthesis and things like that they're going to use to power the source of making their energy. They don't need to bring in food to power their machines. They get it from the sunlight. So that's a photoautotroph, an organism that uses its own energy, energy from the sun, and then it makes its own food, photoautotroph. And you can also have photoheterotrophs, which use light from the sun, but they don't bring in carbon dioxide from the environment. They get it from kind of organic nutrients and things that they bring in from the environment, but they still use the light to power that breaking down. And then you can switch down to chemo, auto, and heterotrophs. We are a chemoheterotroph. We cannot make our own food, but we bring in food from the outside in the form of various chemicals and organic things and inorganic stuff that we eat that we're going to break down, powered by energy from our own bodies to make more food. So we have to pay to make our own energy. Photoautotrophs and photoheterotrophs don't have to, because the sunlight does it for them. So anything that's a heterotroph, you're going to have to pay for your own energy, things like that. So you guys get the idea on that one. It's pretty simple. We've already covered the term fastidious as well when it comes to growing bacteria in a lab. These picky little organisms that need specific growth factors given to them. Things like strep, they won't grow well on nutrient agar plates because it doesn't have enough iron in them for them to be happy and healthy. Whereas if you give them a blood agar plate, which is essentially the same thing as a nutrient agar plate with a bunch of iron added to it, they really like that. So they're fastidious organisms. They need a special nutrient factor, a special thing given to them so they can grow at something termed their optimum rate. Now, an optimum rate is how fast that bacteria can grow at the best overall conditions. So they're happy, they have the best temperature for them to grow at, they have all the nutrients they need, they have everything that they like is perfect for them. How fast is this organism going to divide? And that's what we're looking for to try to achieve that optimum growth rate, giving them the best possible environment that they can have, so they'll be the happiest that they can possibly be and divide as fast as they possibly can for us. So autotrophs. We've already covered the autotrophs pretty well, I suppose. These organisms are not nutritionally dependent on another living organism. They can make their own food. They don't have to eat something else or another living organism to get that food. Something doesn't have to produce the food for them. So we have to eat food. They make their own food. So something had to produce the food for us is essentially what I'm getting at. Photoautotrophs, they can use photosynthesis, as I mentioned earlier. They don't need light, they need light to do this. Some of them don't need oxygen, some of them do, and oxygenic photosynthesis is the requiring of oxygen. You have to have light to be able to break down CO2 and things like that to power your machinery, or oxygen, and then you have anoxygenic, where you don't have to require oxygen to finish the electron transport system, the electron transport chain for energy. So some types of bacteria require oxygen and some don't. in photosynthesis. It's very interesting for these guys. Very primitive form of photosynthesis that bacteria do. It's not really hardcore photosynthesis. It's more like the little predecessor to kind of thing. Very simplistic form of photosynthesis. And then you've got autotrophs. That chemo meaning chemical, auto meaning they make their own food. And they survive totally off of inorganic substances. So they're going to bring in methane, or not methanes, they're going to bring in waters and things like that, weird little substances, and break them down inside of their cells to make their own energy. They get their carbon sources from stripping those carbons and hydrogens and things away from the things that they bring in to their cells. Very interesting. And then you have methanogens, which are kind of chemoautotroph, and these guys can produce methane gas under anaerobic conditions. So if you've ever been around, anaerobic means there's not any oxygen. So if they produce their own energy via antioxygenic photosynthesis, they won't have oxygen. They will produce methane as a byproduct instead of oxygen as the byproduct is regular photosynthesis. So methane, as you know, is a very flammable gas. It smells really bad. And if you've ever been around kind of a yucky, gross, stale pond or a kind of a nasty, stale lake or something, and you've seen those bubbles coming up from the bottom of it, it's usually methane. The bacteria that live down at the bottom of the lake ferment and break down plant matter and things like that in the mud. And as they do that, there's no oxygen down there for them, so they're not being able to use oxygen. So as a byproduct of their anaerobic photosynthesis, or excuse me, their anaerobic chemoautotrophism, they produce methane gas. So it's kind of an interesting little thing. You can actually collect it in a jar and catch it on fire sometimes. It's kind of cool. So the other side of that are heterotrophs, and these are organisms that are requiring food from some other living organism, some other source. So we have to eat food that something else has produced for us. So we can't make our own food. We eat the food that thing that produced the food made. So we're essentially food thieves. We go steal it from something else. And there are two categories of heterotrophs when it comes to bacteria and things like that. You have the regular heterotrophs, the chemoheterotrophs, that break down food regularly, just like we do, and that just do regular aerobic respiration. They require oxygen, just like we do. Traditional metabolism, the exact same thing that we do. And then you've got two other categories of heterotrophs when you're talking about bacteria and some other organisms like fungus as a whole. You have saprobes and parasites. So a saprobe is an organism that feeds on dead, decaying matter. They're free-living organisms. They don't need colonies. They don't need to live inside of another organism or anything like that. And then you have, and they're able to grow on this dead, decaying matter, and they break it down kind of like a spider. If you're familiar with how spiders eat, they catch their prey, they inject the venom inside, and then the venom breaks down the juices inside of the animal, turns it into juice, the little bug, and then the spider just sucks the juice up. This is essentially kind of what they do. They secrete digestive enzymes, these little bacteria. They secrete digestive enzymes. Fungus do this as well. Outside of their cells, it goes out into the environment. It breaks the environment down, and then the bacteria just kind of suck everything back up. So they get a nice digested soup. It's a very interesting way of acquiring nutrients. So a bunch of bacteria do this, fungus do this, things that feed on decaying matter, microorganisms that feed on those dead animals and rotting plants and things like that, also known as detritus or detritus, depending on how you want to say it. And then you have parasites. And these are going to be things that derive their nutrients from another organism directly. So we usually eat dead things or vegetables and things like that. Well, parasites don't. They're going to live inside of another living organism or on that living organism, and they're going to feed directly on that living organism while it's alive. So they're going to be causing damage to that organism as a whole. So they're going to be stealing nutrients from the organism that the organism itself would need. So you're probably familiar with quite a few parasites, things like ticks, mosquitoes, tapeworms, pinworms, things that live inside of us and other organisms that derive their nutrients from us directly as an impact to us. Now, when you're talking about parasites and saprobes and things like that, overall, you have two different types. You have opportunistic and facultative. So things that are opportunistic mean that they're generally not going to cause problems in humans or animals overall. They're going to, if you come in contact with them within the environment, if you touch them, they're probably not going to cause you any problems. But if you have a small cut on your hand, or maybe you're sick already and your immune system's slightly compromised, you can come in contact with these organisms, and if you were perfectly healthy, they wouldn't cause you any problem. But since you slightly damaged your skin, you have a small break, the bacteria or whatever it is might be able to get inside. So when the opportunity presents themselves, they will be able to make you sick. But under normal, healthy circumstances, they won't. So that's something considered an opportunistic pathogen. If the opportunity, the door opens itself, they will walk on in. But other than that, they can't open the door themselves directly. And then you have things that are considered obligates. And obligates are things that are going to make you sick all the time. Things like the flu. If you come in contact with the flu and it gets inside of the right spot in your body, you're going to get sick. It's just how that one works. You get enough of them in there, you're going to get sick. You come in contact with it again, you're probably going to get sick again. That's just kind of how that one works. There are bacteria, there are viruses that are not meant to live out in the environment and chill and not cause us problems. They are directly evolved to cause harm to the organisms. They need us to transfer around from place to place. They need to be causing sickness as part of their survival strategy. So without something being sick, without causing damage to us, they would not be able to spread around. They can't survive very well in the environment, so they have to rely on us to do it for them. So that's something considered an obligate pathogen. So opportunistic versus obligate pathogens. Opportunistic, if you get yourself cut or you're sick already, they might come on in. Obligate parasites, you come in contact with them. Obligate pathogens, you come in contact with them, they're going to make you sick pretty much regardless. And that's the difference between the two. So when we're talking about growing bacteria in a lab, we've talked about nutrition, what nutrition is, how bacteria acquire that nutrition. So now we're going to talk about how we actually grow them in a lab. How we give them the proper nutrition, how we measure their growth, and things like that. Well, bacteria, as I probably mentioned already, can be found pretty much everywhere on the planet. You can see down here we have a glacier, and you can see my mouse. It's not on. Let me turn it on real fast. You can see this little glacier down here. You can see this black line running through it. That's not dirt. That's bacteria that lives on the outside of these glaciers. So that's actually bacteria. They live in these freezing glaciers. Down here is the Marianas Trench with these hydrothermal vents. That's pure sulfuric acid that's being pumped down in them. This is the bottom of the ocean, by the way. If you were to go down there, despite the lack of oxygen, the pH is about 0, so that would instantly melt you. And then the pressure would crush you as well. But there are bacteria that are found down here, and they're called baryophiles, bacteria that like pressure, that require this amount of pressure in their environment. Without it, they'll probably die. And then it's probably likely that there are bacteria-like organisms found on other planets. They are found in all these crazy environments in our planet, so it would make sense that they can survive in other planets in these other strange environments as well. Well, given the fact that bacteria grow pretty much everywhere on the planet, they eat, they live in freezing conditions and they eat snow and all kinds of weird conditions. They eat falling, decaying fish matter and things that fall down at the bottom of the ocean with a pH of 0 and tons of pressure down there. If you want to grow these things in a lab, you're going to have to replicate those conditions. It's that zoo animal concept. Happy animals, happy zoo, they do what they're supposed to. Unhappy bacteria, their conditions aren't right, the environment's not good for them, they're not going to grow, they might not even survive. So if you want to study these guys in a lab, you're going to have to make sure that you can replicate these conditions properly, which is not always easy to do. Well, what are we talking about when we're measuring replication? I've said bacteria grow, we want to encourage them to divide. I mentioned the optimum rate of growth, how fast they divide, how fast you can make them divide, and things like that. So what really is bacterial replication? What am I talking about here? Well, bacterial replication is a process called binary fission. Binary fission is about the most simplistic thing on the planet. You have one bacteria that has a little tiny chromosome, a nucleoid inside of it. They duplicate their little nucleoids, so now you have two, and then the little bacteria splits in half and takes one of the little nucleoids with it. Very simplistic process, super easy, really quick for some species. They can do this in about 15 to 17 minutes. They can divide in half again, and then they divide again in another 15 to 20 minutes. And this is very, very simplistic. Now, it doesn't require a mate since it is asexual. And the problem here is that the upsides are it's really fast. It's very quick. You don't have to require a mate. But the problem is that it doesn't increase the genetic variations in a population. So if the very first bacteria that divided was not immune to an antibiotic, the rest of its progeny and the rest of those from there on out won't be either. They're all going to be identical. So if something comes in, a disease or a new nutrient that one of them can't use, the rest of them are going to be impacted by that antibiotic, that disease. None of them can use the nutrient. So they're all going to be the same. Whereas sexual reproduction shakes up genetics a little bit, genetic variation. Somebody might be able to survive better, use that nutrient, so the species overall might persist. Whereas asexual reproduction, everybody's going to be pretty much identical, and then bad things happen. But the upside for us as a microbiologist is since this is the way they divide, everybody's going to be genetically identical, and that's what you're looking for in that colony concept. You have one little cell that you isolate away from everyone else. It starts to divide via binary fission, and then you end up with a bunch of identical cells that are all the exact same, that you can use for testing and things like that, diagnostic purposes. So when we're talking about growing bacteria, the very first thing that we're going to have to talk about is something called the generation time. How fast do bacteria replicate? How long do we need to wait before we get a new generation? So if we start with 2, how long is it going to take till we get 4? How long is that going to take till we get 8? How long is that going to take till we get 16? And you can figure this out based on a little formula, which is, right here. I'll go back a couple of steps, a couple of slides. So this is the generation time calculation right here. You take the number of cells that you started with, and then you go through this little concept here, and it'll give you the number of the outside cells. So this is a little example. A Taco Bell manager accidentally inoculated 5 cells of E. coli into the burrito meat. I love how it's Taco Bell. How many cells would there be after 4 hours if E. coli has a generation time of 20 minutes? So you plug in 5 here. 20. You can take 4 hours divided by 20. That'll tell you how many generations you have. You plug that into there, and then it'll tell you the number of cells after you go through here. Really easy concept. Really easy way to figure out how many cells that you have after a certain period of time. Now, generation times, they've all been established for pretty much every bacteria on the planet, and you can look them up. They vary from species to species. But for the most part, they're going to stay around the same number of hours and things like that, number of minutes and whatnot. So you can figure out very quickly how long, what the amount of E. coli or contamination to a sample might be through one of these little calculations. There are tons of things that will influence how quickly bacteria grow, and it's not just chilling out like this. We'll talk about a bunch of those things in a second that influence how fast bacteria grow and how slowly they grow and things like that. Well, when a bacteria goes to divide, they're going to go through something very standardized, something called the bacterial growth curve. And this is the bacterial growth curve. If you take a brand new species of bacteria, or a brand new sample of bacteria, and let's just say the Taco Bell guy, let's walk through his little example. The Taco Bell manager accidentally introduced 5 cells of E. coli right here. And this is the introduction into that burrito meat right there. Well, there are 5 cells in there. Let's figure out how long and how fast these bacteria grow using that generation time calculation. But every single bacteria on the planet, for the most part, is going to go through these phases of growth when they encounter a brand new person, a host, or a brand new petri dish, or a brand new environment like that burrito meat. So what's going to happen is they're going to be introduced right to a brand new sample of something right here at the lag phase. And they're going to have to figure out, these bacteria, microbes, whatever they are, that what's going on in the environment. Do I have the right nutrients? Do I have the right pH? Do I have the right temperature? Is this environment good for me? What do I have to use here? And during that time, there are no cells actively dividing. The cells that are there, the number of those cells is not going to increase. You can see that here, the number of cells over here, and time passing. So the number of cells And that burrito meat from 5 has not increased at all. They're figuring out what type of nutrients they have to do. They might be turning on some proteins to process something that they might not have been using earlier. All kinds of stuff they're figuring out. If they're in a brand new human or a brand new host, they're navigating to the proper place to cause infection, avoiding the immune system, and things like that. Well, eventually what's going to happen is those organisms will reach a point where they've figured out what's going on. And you can kind of see that right in here, where they start to divide just a little bit. And this is entering something called the log phase, or the exponential growth phase, the exponential phase, growth phase. There's lots of different names for this. And you can see here that the number of cells is increasing at a very sharp angle. So these organisms, and they have a lot of food for them available in that burrito pan, burrito meat. They have tons of burrito meat. There are only 5 of them to start with, so they have way more food than they could ever need. So they're going to eat and eat and eat and eat and eat, divide, grow, divide like crazy. And the number of cells is going to go up very quickly in a very short amount of time. And then what's going to happen is eventually they're going to reach something called carrying capacity. And this is where they enter into something also called the stationary phase. They hit the carrying capacity, and then they enter into the stationary phase. So what is the carrying capacity? Well, every environment on the planet, regardless of how big or small it is, a petri dish or the entire world, has something called a carrying capacity. And that's how many organisms that the environment can hold without there being detriment to the environment or the organisms as a whole. Now, if you have a fish tank, you're probably familiar with this concept. You're supposed to have, I think it's 1 fish for every square foot of tank or something like that, or half a square foot of tank, and it varies depending on the species. But if you stick, 15 goldfish in a 5-gallon tank, they're going to be swimming on top of one another. They're going to be bumping into one another, and nobody's going to have enough room to move around, which means that the nutrients inside of that tank is going to get, you put a lot of food inside of there. They're going to eat it up very quickly. They're going to produce tons of waste. The waste is going to overpower their environment very quickly. And they're not going to have enough room to move around. If they reproduce, that's going to create more problems with crowding and things like that. And that's called the carrying capacity. So if your fish tank can hold 5 fish, 4 is the perfect amount. Nobody's going to be fighting. Everybody's going to have the right amount of nutrients, the right amount of space. You can maintain the waste. But if you add 1 more fish or 2 more fish, the space is going to be decreased. The waste production is going to be increased, which means that the environment and the animals that are in it, the organisms that are in it, are going to have stress put on them, which causes them to die. And that's what carrying capacity is. So once an environment tends to reach the carrying capacity, organisms are being birthed and dying at the same rate, just to kind of keep that stable. You don't want to go over that carrying capacity line. The environment, you will for a second, but the environment levels it out pretty quickly, and then you stay stable. So the birth rate and the death rate are going to stay the same. The population is not going to grow, and it pretty much stays stationary for a while. It's not going to continue to increase. So then eventually what's going to happen is you're going to run out of food. If you're in that burrito meat, let's pretend that they don't ever throw away the burrito meat, or they just set it out back in the pot and let it sit there forever. The bacteria that are in it will eventually eat their food. So they'll run out of food. Or if they're in a new host, a new human, you're going to start to kill your host. Your host is going to die. So your host dies, which means that your food is now going to run away. Your food's going to decrease. So you can't go on forever because eventually you're going to run out. Now, as the food source starts to run out, they run out of bacteria. They run out of food for the bacteria. There's a whole bunch of bacteria and not enough food for them anymore. So the population's starting to decline as organisms start to starve to death because there's not enough food for them anymore. As the food just gets smaller and smaller and smaller, less and less bacteria can survive because there's not enough food for them. Or in a host, the host is dying or dead, and the body is decaying, the organisms are eating that away, and then eventually the body's going to be gone, and the little burrito meat will be gone, and then there will be no more food for these organisms, and they will die completely. So that's entering into the death and the decline phase. So every single bacteria on the planet is going to pretty much follow the same sort of growth curve when they encounter a new environment or a new host or something like that. So you can manipulate this around by changing the growth conditions. If you want them to grow faster, you can grow them, increase the temperature, which will make this go shorter. You want to slow it down, you cool it down, and it'll make this a little straighter where they're not growing as fast. And you can manipulate this around. And you can also use the standardized bacterial growth curves to kind of predict how fast infections are going to grow, how fast bacteria are going to grow on a petri dish, and things like this as well. So very, very, very interesting uses for these growth curves. So what can you as a microbiologist do in a lab or in the environment? What does the environment do that can manipulate the ways that bacteria grow, and how can you take advantage of this in a lab? Well, there are tons of different ways that we can manipulate how fast or how slow bacteria grow. Temperature, giving them different types of nutrients, adjusting the pH, salt concentrations of medias, giving more or less oxygen, the presence of CO2. There's tons of different ways to manipulate how fast bacteria grow. And you want to be able to understand how these things influence how they grow and how fast they divide if you're going to work with them. So you want them to grow faster or slower. Maybe you're going out of town for the weekend, or maybe you are trying to figure out if you can control an infection by temperature. So you want to see how fast they grow, and you want to know how fast they grow so you can see if your experiments manipulate or increase or decrease how fast they replicate. So very useful to understand how these factors influence how quickly things divide. When you're talking about growing bacteria, really the first main thing you need to be concerned with, other than giving them the proper amount of food, is the proper temperatures. Just like us, we have a temperature range that we really like to be living in. Anything hotter than about 95 makes us sweat, it's very uncomfortable, and anything lower than about 65 makes us shiver. And we would really like to be chilling at about 70, 75, somewhere around in there. For most people, that's our body temperature, room temperature, where everybody seems to be happy and content. We function best at that temperature. Anything colder than that, our body's going to shiver. Super cold, our body will shut down. We will eventually freeze to death and die. Anything hotter than that, our temperature is going to rise on the inside. It'll bring our body temperature up. We can't sweat enough to maintain our body temperature, and eventually our proteins and things inside of our body will start to break down because of that severe heat. And bacteria have the same problems. If you're going to grow them in a lab, you have to be aware of these three things here: the minimum, the maximum, and the optimum temperature that you can grow bacteria at. Now, the optimum temperature promotes the optimum growth rate, hence the name. Most bacteria that you work with in a lab are going to be grown somewhere between 25 and maybe 55 degrees Fahrenheit, or Celsius, and the majority of them are going to be grown around 37, somewhere around in there, because that's human body temperature, 98.6 degrees, so 37.5, somewhere around in there degrees Celsius. And that's the optimum growth temperature for many bacterial species that live inside the human body. That's where they're naturally going to be found, so we replicate that environmental condition in a lab incubator. So anything cooler than 98.6, 95, 80, something like that, they're not going to be dividing as fast as they could. Anything hotter than that, they're not going to be dividing as fast as they could. They're going to have to be spending more time regulating their temperature, cooling themselves down, repairing broken proteins and things. So they're not going to be concerned with reproducing. So they're going to not divide as fast as they could. Cooler than that, they're going to be shivering. It slows down metabolism. It slows down the movements of proteins and molecules and things. So they're just going to be functioning slower overall. And then on the other hand of that, you have the minimum and maximum temperatures that a bacteria can grow in. The minimum temperature is the lowest temperature that you can expose a bacteria to without it, and the growth of that organism and metabolism will cease. Now, the bacteria itself might still be alive. It won't be dead. It just won't be able to grow or produce more energy or divide. It might just go to sleep, essentially what's going on. It goes kind of into hibernation phase. Now, it'll just sit there. It won't grow. It won't make any energy. It'll just sit there. That's the minimum temperatures. The lowest temperature you can get to where metabolism still happens, where bacterial growth still happens. Anything lower than that, they're going to stop. You can't go lower than that minimum temperature, or else they'll just go into hibernation, or they might die. You might kill them. So the minimum temperature is useful if you want to see how long it takes something to grow really slow, or you're trying to figure out if temperature really does influence growth rate. Or maybe you want something to be able to sit in the incubator over the weekend while you're on vacation, or you have a couple of days for a research project or something. You can lower the temperature on your incubator to the minimum temperature, which will still cause the bacteria to grow, but not as fast as it would if you left it at 37.5. And on the other hand of that, the flip side, we have the maximum temperature. And that's the highest temperature that a bacteria or a microbe can tolerate before it ceases its growth or metabolism. So if you go higher than the maximum temperature, it's going to turn off the growth. It'll turn off the metabolism. You might outright kill them. Anything lower than that is going to be slower than the optimum temperature, but faster than the maximum. So you don't want to go over the maximum. You don't want to go under the minimum. You can work with those if you're trying to manipulate your experiments, or that's all that you have. But the optimum temperature, if you have the ability to go for that, is the best thing you can find to grow bacteria. Now, there's charts out there for optimum temperatures of growth, or if you're getting your environmental sample, you need to replicate where it was found. So if it's a human-dwelling organism, replicate human body temperature. If something dwells in a cat, they're a little warmer than us. Or if it's something that dwells in the Arctic, you need to replicate those conditions in that lab for that optimum temperature. Now I'm going to go ahead and skip just a little bit of this, because this is the same thing that I'm going to talk about. These are the word forms of this little chart. So I'm going to talk about the chart because it's easy to go over, but the word form of that, if you want to read them, are before this. So this is the same information, these couple of slides, that's found in this graph. So I'm going to go over it in graph form, but if you want to go back, you can read that yourself if you'd like. So this is the bacterial temperature classifications. So these are what microbiologists use to classify groups of bacteria into different types of temperatures, or different temperature classifications, different groups, if you like. So let's start over here with the cold ones, and over here with the hot ones we'll work our way to. So right over here you can see how fast they grow, the rate of growth, and the temperature in Celsius on the bottom here. Now, how do you read this graph? Well, right here, this is a classification of organisms called psychrophiles. These guys live in that Arctic environment. They grow on those glaciers. They love living in cold environments. They are adapted to that. They're evolved for that. So if you want to grow these types of organisms in a lab, you have to replicate that type of environment. So what do we see here? Well, right here is the minimum growth temperature for the group of organisms classified as psychrophiles. Over here is the maximum. And right there in the middle is the optimum growth temperature for psychrophiles. Right there, about maybe 12 degrees Celsius. So right there, if you want to grow psychrophiles in the lab, their optimum growth temperature, if your organism is classified as a psychrophile or you pulled it out of a really cold environment, chances are the optimum growth temperature for it's going to be at about 11 to 12 degrees Celsius. So set your incubator at that, and these types of organisms will be quite happy. Now I'll skip over Heterotrophs for a second and move on to mesophiles. Mesophiles are, hence the term meso meaning middle, organisms that are found in the middle temperature ranges. So their minimum growth temperature is about 10 degrees Celsius, and their optimum or maximum their maximum is about 48 degrees Celsius. The middle here is about 37.5. Their optimum growth temperature sounds familiar, right? Human body temperature. So mesophils are organisms that tend to cause problems in humans, live on other warm-blooded animals, and things like that. So mesophils are the vast majority of organisms which are going to cause problems in living organisms because they grow, their optimum growth temperature is right around our body temperatures. So mesophils are the ones that are going to be causing problems with humans and animals and things like that for the most part. Thermophils, or thermophiles, I should say. These guys have a minimum growth temperature of about 39 point some odd, and about 71 is their maximum, and their optimum is about 62. These guys are going to be found living out in the sun, in the desert, and things like that, where there's a lot of heat. They're a little more desiccation tolerant. They have to be able to survive that higher temperature. And then you have your hypothermophils. And these guys live all the way up past 110, which is boiling in Celsius. Their optimum growth temperature is quite wide because these guys can be found in those deep sea thermal vents where it's really, really, really hot and almost boiling down there or over boiling. And these guys can survive in these crazy environments down there, and because their cells are adapted to survive in that. So this is the temperature classifications for bacteria. If you know what type of organisms you're working with and they're classified as one of these guys, you can go ahead and set your incubator at the optimum temperature based on these graphs, and this is probably going to be the best way to grow them in your lab. Now, Psychotrophs are a weird little group. They're technically considered mesophiles, but they can grow quite well in the refrigerator or in the incubator. Now, they really don't mind being in the fridge. They really don't mind being in room temperature. They kind of can grow in the middle. They're strange little organisms, so just kind of gloss over them for a second. So, already talked about nutrients. Sorry, come back. And pH. So pH is another big thing that can be used to manipulate how fast bacteria grow in a lab. And when you find bacteria in the environments, there's a wide range of pH that's found even inside the human body. So things like 0 or 1 in our stomachs, and then basic, pretty much the rest of our body and some other places and things like that are neutral for other places. So bacteria, when they live in different environments, even on our body, so imagine what the whole globe is, they have to be adapted to survive in those little tiny environments that they're found in. So if there's a bunch of acid there, the bacteria that can survive there have to be able to survive in that high concentration of acid. Sometimes they might even have to have that to survive. So if you live on, you say, your skin and you get inside the intestinal tract or the stomach, that acid might kill you. So you need to be aware of that as a microbiologist when you're going to grow bacteria in a lab. Do these bacteria like acid? Or do I need to figure out if they like acid and try to add the acid to them and remove it or take it away and see how they grow best? So pH really does play a large role in growing bacteria at their optimum rates. In fact, there are lots of medias that have been developed that manipulate pH to kill particular species that are non-tolerant of acidic environments or basic environments. When you're looking for a particular species of interest, you can kill off the ones that can't survive the acid if you know the one that you're looking for can. And you can get a very quick little sample to figure out if what you're looking for is there, just killing off everything based on pH. So very interesting use of pH. Salt conditions. You can manipulate the amount of salt in the environment, the amount of salt in the media that you're making, and things like that. And there are lots and lots of bacteria that really like salt. Some require high concentrations of salt. And then there are some that just can't tolerate salt at all. So you have bacteria that live on our skin. In our skin, if you've ever sweated, you get really salty. If you've ever licked your hand after you sweat, it's super salty. So our skin is covered in high concentrations of salt. And that's to prevent bacteria growing on our skin. Well, there are tons of species of bacteria that live on our skin as well, but they are adapted to live in that high concentration of salt. Pathogenic organisms aren't. They tend to live inside of us. For the most part, they're not. If they land on our skin, the salt's going to dry them out, just like a ham. So this salt's going to suck all the moisture out of them and they will dry up. So salt is a really good way to manipulate how fast bacteria grow or keeping them from growing and things like that. If you think about the environment, there are places out there that are full of salt, like salt flats and things like that. And there are organisms that live on these dried out oceans, dried out salt lakes that are just literally the pure salt. And these organisms are called halophiles, salt-requiring organisms. Halos, salt. So organisms that require salt. And then there's organisms that live on our skin that they don't really have to have that amount of salt. Too much salt can kill them, but they don't mind a lot of salt. It's not going to be bad for them. Too much can kill them, but they don't mind a lot of it. And those organisms are called osmotolerant. It's not going to hurt them. It's not going to help them. It's not going to do anything for them. They don't need the salt, but they can stand the salt concentrations. Whereas an organism that's considered a halophile, if it does not have a very high salt concentration, it will die. It has to have that salt for it. And that's the difference between the two. So organisms that are found in the environment, for the most part, that are going to be making us sick, they land on our skin. Salt's going to dry them out and they're going to die. Whereas the organisms that live on our skin, they can tolerate that salt. It's not going to dry them out and they're not going to die. So this is what happens with salt. it's the simple diffusion concept where the water inside of the cell is going to rush out of the cell to try to dilute the salty environment around. And the cytoplasm inside the cell is going to shrink away from the cell membrane, the cell plasma membrane, and shrink up into a little ball inside of the middle of the cell wall, and the cell will eventually die. And it's called plasmolysis, the loss of water to the outside environment. So these high salt concentrations are going to cause this to happen inside of a cell. Now, if you're osmotolerant, your cell can handle this. This water is not going to happen, or this loss of water is not going to happen. But too much salt will still cause this to happen. You can tolerate a lot, but not too much. Halophiles, their cell membranes and cell walls are meant to handle as much salt as you can throw at them, and without that salt, they won't be able to survive at all. Whereas most bacteria, as I mentioned earlier, they're going to be making us sick. They land in a high salty environment, and this is going to happen to them almost instantly, and it'll cause them to die. So we know this. And we can manipulate this in a lab using different types of salts concentrations in our medias. In fact, there's a media that's called mannitol salt agar. You might recall that from an earlier lecture, where it's literally just 7.5% salt. That's almost halfway to a country ham. That's really salty. In fact, that's like way higher than the concentration of salt in blood, which I think is like 0.09 or something. So really low, the saline concentration of blood. So super, super salty. Staphylococcus bacteria that grows on our skin, it's supposed to grow on our skin, they're osmotolerant. They grow on that perfectly fine. Pretty much every other organism is going to die. The salt will kill them. So it's a very quick way to test if you have staph present. Just take a sample from your patient, streak it on a mannitol salt agar plate. Everything that can't survive in the salt dies, which is mostly going to be staph. And if you have staph, it will grow perfectly fine, and you can see it there. So salt is a very useful tool in microbiology. And oxygen. Now I'm going to skip over this one once again. Wrong thing here. We'll talk about this in a second, and then we'll talk about how to wrap this up at the very end with oxygen. So oxygen. Now oxygen is probably the most important factor that influences bacterial growth, much like us. You can survive without food for 3 weeks, you can survive without water for 3 days, but you can survive without oxygen for about 3.5 minutes, and that's pretty much it. So oxygen is going to be way more important for organisms as a whole than other types of nutrients. So the proper amount of oxygen in the environment is very important for bacteria to grow properly. Much like us, they use oxygen as their terminal electron acceptor, some of them do, and they have to have oxygen to make more energy. No oxygen, what's going to happen is when you use oxygen as your terminal electron acceptor in the electron transport chain, you build up toxic byproducts by doing that. So you strip away the oxygen, and when you strip away the oxygen for your electron transport chain, what's left behind are things called free radicals, or singlet oxygens, peroxides, hydroxide radicals, superoxides, strange little molecules which are damaging to the cells. Radicals, they will stick to each other like magnets from across the cell. So if half the cell is on one side of the cell and the other radicals on the other half of the cell, they'll just stick to each other through the middle and rip up DNA and anything that's in the middle. They'll just stick to and rip apart cell walls and cell membranes, so they'll just kill cells. So all these toxic byproducts that are built up by the process of using oxygen for metabolism build up inside of the cells of us as well, but bacteria too. So we and bacteria as well have to get rid of these toxic byproducts of oxygen, or else it'll build up inside of the cell and kill it. So bacteria that live in and require oxygen inside of their metabolism, that require oxygen, have ways to get rid of these toxic byproducts. They've developed enzymes and things that will neutralize peroxides, things like catalase, and neutralize these free radicals and get rid of them for them so that it doesn't kill them. Now, not every microbe, bacteria on the planet is capable of using oxygen, is capable of dealing with these toxic byproducts of oxygen. Now, if you bring in oxygen, that's kind of an unfortunate side effect of living. You bring in oxygen kind of regardless of what type of cell you are. So bacteria that don't have the ability to break down these byproducts of oxygen, if they come in contact with oxygen, they still bring it in. And they will instantaneously make these byproducts of metabolism, but they don't have a way to get rid of them. So it builds up inside of their cell, so it kills them. They can't break it down. So if you're one of these unfortunate microbes that has this problem, you have to live in an oxygen-free habitat or a very oxygen-low habitat, or else these byproducts will build up inside of your cell and kill you. So there are tons of bacteria that require oxygen, that can live in oxygen because they can break down these byproducts of metabolism. And then there are tons of bacteria that cannot live in the presence of oxygen and do not require it at all in their metabolism because it'll kill them. And that's kind of the unfortunate side effect of how that one works. So let's talk about the different classifications of how bacteria deal with oxygen. Well, the first one is going to be something referred to as an obligate or a strict aerobe. Things that require oxygen in their habitat to grow and thrive. We are an obligate strict aerobe. Without oxygen, we will pretty much die instantaneously. And the same thing works for these guys. These types of microbes, without high concentrations of oxygen in their environment, they will not be able to produce energy. Their metabolism will back up. They will have a backlog. No energy will be produced, and eventually ATP will run out inside of their cell, and that organism will die from the lack of cellular energy. So oxygen is the terminal electron acceptor. They can't get rid of any electrons via oxygen, since there is none, and they die. Now, this is super useful knowledge because knowing this, you can figure out where on the human body these types of organisms are going to likely cause infections. If something requires oxygen in its diet or in its nutrients, in its life cycle, where on the human body is there a lot of oxygen? Well, is it going to be deep inside of my muscle tissues or deep inside of my internal organs? Probably not. But is it probably going to be on my skin where there's tons of oxygen, or inside my respiratory tract where there's tons of oxygen? And the answer to that one's probably yes. So this is an organism called Pseudomonas aeruginosa, a very common bacteria. If you've ever had green phlegm, it's kind of one of the bacteria that's associated with that. When you get a cough, you cough up that lime green kind of yucky phlegm. It's one of the bacteria that's associated with that. Very common organism in the environment. Very strict obligate aerobe, absolutely requires large amounts of oxygen in its environment, and it produces infections in the human body, in the respiratory tract, on the skin, on open wounds and things like that, not on the insides of humans, because it needs that high concentrations of oxygen to be able to survive successfully. On the other hand of that, we have an obligate or a strict anaerobe, organisms that cannot be in the presence of oxygen at all. It might kill them, or it just turns off their ability to function. So these are these guys that can't deal with the breaking down of oxygen byproducts. So if they come in contact with oxygen, they'll bring it in. The oxygen byproducts will instantly be built, those toxic byproducts, and then they have no way to get rid of them, and that organism will die. So that's these types of organisms. So they use something other than oxygen, some sort of chemicals and things as their terminal electron acceptors to make energy. They cannot use oxygen, or they don't, to do that. So these guys are going to usually be found in the bottoms of lakes and rivers and things like that, mud, dirt, and things like that, where they can get away from the oxygen. You have to think about the fact that a bacteria is this big. If you live in a dirt clot that big, it's a pretty good chance the oxygen is not going to get to you. So lots and lots and lots of these organisms live in the dirt. They live underneath the water where there's not a lot of oxygen anyway, and things like that. Now, one of these that you probably are familiar with is tetanus, Clostridium tetani. This is also called broke-back fever, lockjaw, and things like that. It's the shutting down of the body's ability to move. Essentially, you just turn into a rigid little... you do this, all your muscles tense up, you can't relax. Eventually what's going to happen is your jaw will lock and clench, you can't open your mouth to eat anymore. Eventually what would happen is a long time ago, they'd either break your jaw open and force feed you, or they'd knock your teeth out and poke food through your mouth that way. And then eventually what happens is you're either going to get over it and survive, or your intestines shut down and then your food can't digest and you die that way. It's a very horrible way to die. But tetanus, you have probably heard that you get this from stepping on a rusty nail. Now, that's not true. Rusty nails tend to be found in the dirt. Or you step on a nail, it's covered in dirt, or you step in a nail and you go, ow, that hurt, and you step your foot right back down on the dirt. So tetanus, Clostridium tetani, that bacteria is an obligate anaerobe. It lives in the dirt. The rusty nail just tends to be a good, convenient way to get it to you, to get it inside of you. Now, given the fact that it is an obligate anaerobe, it can't get through you on your skin. It has to be poked inside. And that's why those penetration wounds are so important for tetanus and things to get shots, because any dirt that's shoved up inside of your body, it's avoided the oxygen on your skin. It went way up deep into the muscle tissue. There's not a lot of oxygen, if any, there anyway. So that organism went from an environment that it liked into another environment that it's liked, that's got lots of nutrients and lots of warmth. So that's how that transmits. It's not the nail itself, it's not the rust, it's the organisms that live in the dirt around that rusty nail. Then you have facultative aerobes, and these guys don't care one way or another if oxygen is there or not. Facultative anaerobes. They don't care if there's oxygen or not. If it's there, they're going to use it. If it's not there, they're not going to use it. They don't care one way or another. If it's there, they're going to use it. They're going to grow a little faster than they would without it. But if they don't have it, who cares? Not a huge deal for them whatsoever. They might just grow a little slower. It's kind of like if I had a Porsche, I'd be really happy, but I drive a nice car anyway, so I'm cool with it. Kind of that idea. So you just go a little faster. And that's pretty much how these types of organisms work. Bacteria that live in the intestines, yeast, and things like that are very common facultative anaerobes. You swallow a lot of oxygen when you eat sometimes, or when you drink. And if they get a lot of oxygen in their intestines, awesome. If not, and not a huge deal, they don't need it. They can survive without it. But if they got it, cool. Microaerophilic organisms. As the name implies, micro meaning just a little bit. And this is the Goldilocks bacteria of oxygen affiliation. This guy likes just a little bit, but not too much. And they usually like a little bit more carbon dioxide in the environment than you're usually going to find just in the concentration of oxygen, just pure air itself. So these guys, when they invade a host or in the environment, they will move around and navigate themselves to find the right amount of oxygen that they like. So just like Goldilocks, she went around and went to different beds until she found the one that was just right. These guys will do the same thing. They'll move around in the environment until they find just the right amount of oxygen for them, and they will stay in that one little location. They won't move around too much. Helicobacter pylori causes stomach ulcers. Is one of these types of bacteria, and it burrows into the intestinal and stomach lining to get away from the oxygen in the stomach. So it'll burrow in to the point where there's not enough oxygen, but or not too much, but just enough for it, and that's where it stops, and that's why it does that. So fun little bacteria. And then you have aerotolerant. These guys don't care if oxygen is there one way or another, because they just can't use it. They don't care. If there's oxygen present, it doesn't matter. It's kind of like if I had $1 billion Brazilian dollars, Confederate dollars or whatever, it wouldn't matter, because there's no such thing as the Confederacy, and I don't live in Brazil anymore, or ever. So that money is useless. Oxygen would be useless to these organisms. They just can't use it. They can't do anything with it. They're just said to be indifferent to the presence of oxygen or not. This just doesn't exist for them. They're not even aware of its presence. So these guys, they tend to like a little higher levels of CO2 in their environments. So Streptococcus pyogenes is one of these, called a strep throat. It sits in the back of your throat. You exhale a lot, so it gives it a lot more CO2. It doesn't get a ton of oxygen coming in. But it gets a lot more CO2, and the oxygen that does come onto it, it doesn't care, because it doesn't use it, it can't use it, it just doesn't matter to these types of organisms. So when you grow bacteria in a lab, you have to take into account all of this stuff. So how do you figure this stuff out? Well, it's super easy. We'll come back a step to that other slide. This is how you figure this stuff out. It's super easy. It just takes a test tube and some broth. So what you're going to do is you're going to take a sterile test tube with some broth in it, about half full, and you're going to inoculate it with a small sample of your bacteria. So let's pretend you don't know what type of bacteria you have, how it's going to deal with oxygen, if it likes oxygen at all. And you're going to inoculate it up at the top, and you're going to incubate it for about 18 to 24 hours and then come back and look. And the bacteria will tell you how they grow and how they require and use oxygen, or if they do at all. So the very first one you can see here is something called an obligate aerobe. We already talked about that one. These organisms require oxygen in there for their metabolism to function. And you can see here that they've all gone up to the very tip top of this tube. They are all gathered up at the top where there is as much oxygen as they can possibly get in their metabolism. There's none down here at the bottom where there's no oxygen. They're all gathered up at the top. They have to have oxygen, and you can see that because they're all trying their best to get as much as they possibly can. Now you come over here to a facultative anaerobe. These guys don't have to have oxygen, but they prefer it, and that's kind of how that one works. So you can see that here. They're gathered up all at the tip top. There's a lot more of them there, but they are also dispersed throughout the rest of it. They like oxygen, they would prefer to have it, but if they don't have it, it's also not the end of the world, so they'll grow down there as well. And then on the other hand, you have our obligate anaerobes, organisms that cannot live in the presence of oxygen. It might overall just kill them. They can't break down those toxic oxygen byproducts, so they have to do their very best to get away from all of that oxygen. So you can see here, they're down at the very bottom, as far away from all the dissolved oxygen as they can possibly get so they can still survive. You have aerotolerant. These guys don't care one way or another. Oxygen doesn't matter to them if it's there or not. They could care less. You can see they're equally dispersed all the way through it. Does not matter to them if it's there or not. Then you have microaerophils, and this is the Goldilocks one again. And they're going to navigate to the area of the tube that has the right amount of oxygen for them. And then they're going to stay pretty much in between that little too much zone on the bottom, or too much on the top and not enough on the bottoms. And then they're going to navigate and kind of stay and grow in between that area. So they will tell you what type of oxygen affiliation that they require. Now I've mentioned, and if you know what species it is, that will already be way established for you ahead of time. Carbon dioxide. Now I mentioned that some organisms require large amounts of carbon dioxide, or they like it, and these organisms are called captanophiles. And there's a bunch of different ways to fix or to give CO2 to organisms. You can put them in a CO2 chamber, which is what this is. They have round ones, they have squares. You can pump in CO2 gas directly, but this is one of those common ways. And this is called a gas pack or a CO2 canister chamber or something like that. And essentially what it is it's an airtight box. You put a capthanophile, organisms that like a little more CO2 in their environment than we need. Everybody has to have it a little bit. Some of them like way more. So you put them in this little box. You click it down on the sides. It's airtight. It's got a rubber seal around it. No air is going to come in. And then you put in a little, it's kind of like one of those heat packs that if you're familiar with the little thing you pull out and you put it in your hands, the little packet of little thing. It looks kind of like that. You put that in here, and you can see them right there on the side, one there, and a little bit of water, and it's going to produce a ton of carbon dioxide. It'll fill that chamber up with carbon dioxide, and those little organisms will be very, very happy growing in there. So carbon dioxide is a very cool, somewhat strange growth factor. Okay, so now that we've grown these bacteria up in a lab, it's important to figure out how much we have. How fast did they grow? Did we get enough? So are there a bunch of bacteria in this sample that I collected from the environment? Is it healthy for these people to swim here? Is it healthy to drink? So lots of different methods can be accomplished by counting bacteria that you've grown up in a lab. So there are two different ways to count bacteria. Actually, there's a bunch of different ways. But there are two main different types of counting methods. We have something called a direct cell count and a viable cell count. So a direct cell count is going to be counting bacteria that are alive and dead, unfortunately. So bacteria that are alive, they obviously can reproduce and create more, but dead bacteria can't, but they still get counted. Viable cell counts, on the other hand, only count bacteria that are capable of multiplying. They're capable of dividing, growing more, and increasing the count of bacteria in that sample. So that's the difference between a direct and a viable cell count. So let's talk about some direct counts. A direct microbe count, this is literally what this is called. You take a little slide that has a grid section. It's kind of like notebook paper or graph paper. And you can see that down here. And you take about a milliliter. So you get a sample of the environment or a sample of your petri dish or whatever. And you take about a milliliter. So say you got 300 mils of sample. You take a milliliter out. Put that 1 milliliter on your gridded slide. And then underneath your microscope, you sit there and you count 1, 2, 3, 4, 5. Every single one of these little teeny tiny bacteria. And then what you do is you went, well, I had 400 bacteria in a milliliter. I have 300 milliliters. What's 400 times 300? And then you figure out the whole concentration of bacteria in that entire canister, in that entire sample that way. Now, this does not count or tell the difference between living and dead bacteria. You can't tell the difference between a living or a dead organism, a bacteria, underneath a microscope. So, unfortunately, you might be counting a bunch of dead organisms, but you still knew how much was there. And sometimes that matters because dead organisms can have toxins inside of them and still cause problems. So that is not necessarily just a loss, but it requires a very large sample, and you need a very accurate counting method. You can lose your count. There's all kinds of problems with this one, but it's a good way to do this if you have the time. You can use a flow cytometer. And these are super cool, and this is, once again, a direct cell count. You take a sample up at the top, and what's going to happen is it kind of works like a little funnel. You put your sample in a liquid form up at the top, and it's going to have a very small little output that will control, like 1 cell or 2 cells at a time will only be allowed to go through there. And as the water flows through, there's a little laser beam that passes through the funnel down at the bottom down in there. And as the little cells flow through, they break the laser beam. And every time the laser beam's broken, it counts it as 1. So every cell that goes by, 1, 2, 3, 4, 5. And that's how you tell how many things are in your sample. So super cool again. Really interesting way to do this. But once again, it works on the concept of shadows. So whatever breaks the laser beam casts a shadow. Whatever breaks the laser beam. Living things break laser beams, dead things break laser beams, it doesn't matter. So once again, living and dead bacteria are both going to be counted in this. So if you want to know a really accurate number of how many bacteria are in there overall, good way to do it. If you want to know if they're all dead, not a good way. If you want to know if you have too many living bacteria that can grow and repopulate very quickly, also not a good way to do this. Plate count, on to our viable count methods. So plate count is a viable count method, and this is going to be directly counting colonies of bacteria. So this is super neat. So what you do is you take a sample, so you take your 300 milliliter sample, you take a milliliter out, and you put a milliliter on a plate, and you smear it around with a little hockey stick, and you let it incubate. You come back, you look at it, and you count the number of colonies that are on the plate. Well, there's a ton here. I can't tell how many colonies are on that plate. So this is termed TNTC, too numerous to count. I don't know how many are on this. But what I can do is I can take my original sample, I took 1 milliliter out of it, and I have to dilute that sample, 1 milliliter into 9 milliliters of water, or 1 to 10 dilution, and then do it again. That's still way too much to count, TNTC. So now I got to do it again. Take my original sample, 1 milliliter, into 99 milliliters of water. And that's a little better. And then I got to do it one more time into 1,000 milliliters of water. So you have to do dilutions down till you can get to counting these cells. 1, 2, 3, 4, and you can count the individual colonies now. And now what you have to do is you go, there's this many colonies. Oh, what was my dilution factor? Times 1,000, times 100, times this, times 1. And you have to go all the multiplication back up to the original sample, and then that's how you figure out how many bacteria were in your original sample. So there's some math involving this one. It's a little complicated, but it's the most accurate way to do this because it's only going to count living colonies. If you put a dead bacteria on a petri dish, it's not going to be able to grow, it's not going to be able to divide, so you're not going to be able to count it. It just won't count. Requires at minimum 12 hours to grow petri dishes. And you might have to dilute 3 or 4 of them, which require 12 hours each. So this is not something that you're going to do if you're in a rush. So here's that dilution colony again, or dilution factor again, excuse me. And you can see here, way too many, you dilute it down, dilute it down, dilute it down, and then you dilute it down again. And then you got to do all the math back up to get to your original sample, and that's how that works. So you can see kind of what's going on here. Membrane filtration. You can take bacteria on the sample, pour it onto a filter, pour it through the filter, suction all of that out with a vacuum tube, and then you take the little filter off, put it onto a petri dish, and then incubate it. Only living things are going to grow. Only living things are going to develop colonies. Super cool way to do this. And then what you do is I sampled a 500 ml flask. There are 800 bacteria that grew in that. That's how many are in 500 mils. Maybe my original sample was 6 liters. And you just do some math and you can figure it out from there how much was in the original sample. So super cool way to do this. And then you're occasionally going to counter some things that are a little too big to just overall sample. You can't really count the bacteria or the algae or the contents of things in a lake or an ocean or something like that. And this is going to be where you do things by weight. You're going to take a sample of ocean water, evaporate all the water, and then count the solids that are left behind. And if it's over a certain amount, it'll tell you, oh, this is too much algae to be swimming in, or too much bacteria is present, and things like that, where it's just not feasible to really count the entire ocean. You just need to know if there's too much or too little. So that's what this is for. And this is what they're doing when you see those signs of like toxic bacteria in this area, do not swim, or algal growth bloom in this area, toxin contents, toxin warnings, and things like that. So they're just counting the amount of toxins in a small sample and then projecting it out into the whole ocean. Because if it's toxic right here on the ocean, there's a very highly likelihood chance that it's also toxic over here. So that's pretty much how that works. You can count bacteria using a spectrophotometer. This is, once again, a direct count. It doesn't tell the difference between living and non-living cells. It's the same concept as a spectrophotometer. You break the beams of light, but in this case, you're measuring it all at once. You measure the tube. You measure an empty tube with just broth inside of it. It counts how much light goes through it. You take your sample out, you put your sample in with the same type of broth full of bacteria, and it measures what the difference between the light is. So if 100% of the light went through with nothing in, you can see that here. You put in your tube with bacteria in it, significantly less light went in, and then you can do a little formula to figure out what the difference between this is and what percentage of that is bacteria. So super cool way to do this, but once again, does not tell the difference between living and dead bacteria. And then the final way to do this is mass. You can just weigh them. You can put them in a centrifuge, spin them around really fast, and eventually what's going to happen is the bacteria that are floating around in the little sample solution or whatever will pellet, this is what this is called, at the bottom of the test tube. You weigh the test tube before you put anything in it, so you know what an empty test tube weighs. You weigh it before you spin it with the bacteria and the liquid in it. Spin it, get the bacteria stick at the bottom, pour out the weight, weigh it again, and then you can do some math to figure out what the weight of the pellet is overall. And then you can calculate what the percentage of that pellet is bacteria with some other math formulas and things like that, and figure out how many cells are in that pellet. Super cool way to do this. Super easy, super cheap, super quick, but it's also the least accurate. Dead bacteria have weight. That thing could be all dead bacteria for all you know. Cellular junk, it could be pieces of nutrients that are in the broth that got left behind. It could be anything that gets stuck in that pellet. So this is the least accurate way to do this, but it's the easiest and the quickest. We're going to finish this little lesson off real fast with some relationships between bacteria that are living in the environment. So we've talked about parasites and things like that. How do bacteria interact and grow with other organisms around them? That's the reality of life. The reality of biology is that you will have to interact with other organisms. And those other organisms can either have a positive, a negative, or a neutral impact on how that organism lives as a whole. So if I have good relationships, bad relationships, or neutral relationships, those things will impact how successful I am as a microbe living or doing my thing. So relationships between organisms really do matter. So there's two types of relationships, really, when you're talking about bacteria. You have symbiotic and non-symbiotic. Organisms that live in close relationships with one another that require one or the other one of those groups, or maybe both of them, to be directly interacting. So without one or both of them, that other organism will not be able to survive. And then you have non-symbiotic relationships, which are organisms that don't require other organisms for survival. You don't directly depend on another species of animal to survive, or another species of organisms to survive. Whereas symbiotic, these guys do. They directly require some other organism, a different species than them, to live with. And that's how this one works. So let's go ahead and follow these up real fast. So symbiotic relationships are broken down into 3 different categories. You've probably heard these before: mutualism, commensalism, and parasitism. Mutualism. This is an obligate relationship, which means it absolutely has to happen, where both of the species are dependent on one another to function. This is the concept of, if you're familiar with honey creepers and trumpeter flowers in Hawaii, the beak of the trumpeter the beak of the honey creeper looks like this. The trumpeter flower looks like that, and it's the only organism on the planet that has a beak that's able to reach down inside, get the nectar, and fertilize that flower. They directly depend on one another for their survival. In the microbial world, there are tons of these things where the bacteria, they have to grow in the presence of another bacteria that they either are going to get some nutrient source from, swap nutrients with, or they just need as like a support organism, things like lichens. So weird relationships between bacteria, mutualisms, but they're a little more common in the animal world. Commensalism. And this is when organisms work together, but one of them is helped and the other one is not hurt or harmed. It's just kind of like, if you're familiar with remora fish on sharks, the little things that just kind of stick to them, they don't really do anything for the shark. They don't help the shark out. They don't hurt the shark. But the remora doesn't have to swim around the ocean anymore. And if the shark catches a fish or some prey, the remora can just swim off and catch some, eat the little scraps that the shark knocked off into the ocean. So the remora doesn't have to hunt. So the remora just gets to sit there and do nothing, while the shark provides all the food and movement that the remora fish can need, and the remora doesn't hurt the shark. So that's commensalism. And then you have parasitism. Parasite is directly dependent on the host. Without the host, the parasite will die. Its life cycle will end. The parasite will be over. The parasite is directly benefited. It gets food, it gets nutrients, it gets a nice warm place to live, it gets a reproductive strategies, reproductive vessel, whereas the organism that it's living in is having its nutrients stolen from it. It has something living inside of it, eating it, and may eventually kill it. So parasitism is an interesting form of ecological association. There are quite a few parasites. You're probably familiar with these in the sense of tapeworms and things like that. There are hundreds more of them, though. So then we have our symbiosis, and these are guys that just live together but don't necessarily function. They don't depend on one another. Strange little guys. So our non-symbiotic relationships here. So synergism and antagonism. Organisms that cooperate to produce a result that none of them can do alone. But they don't directly have to have these relationships for survival. They could live without it, but if they help out each other, it's a good thing. And then you have antagonism, where one organism's directly going to impact the survival of another one. So let's talk about those. So synergism would be our leafcutter ants down here. They all work together to form this giant colony of fungus that they will use to eat and feed the queen. They could never do that by themselves, but they work together to do that. And then our other one over here, you can see here, that these fungus are going to produce an antibiotic that kills the other fungus around them. So this is antagonism. They are being antagonistic. They are stopping the growth of another fungus for their own good. I can keep these nutrients. You can't have it because I can keep you from coming in. So that's antagonism. We've already hit these relationships between people and bacteria a little bit with these normal flora concepts, opportunistic pathogens, and things like that. Normal flora on the body are things that are supposed to be living on us, things like staph, things like are in us, things like staph, things like E. coli. There's lots of yeast on us. A couple of protist species live inside of and on us. And all of those types of relationships can be found on or in us as well. Commensalistic relationships where we're not harmed, the bacteria just live on us. Parasitic relationships where the bacteria or microorganisms directly harm us. And then synergistic relationships where they get a nice warm place to live, and they give us a boost to our immune system. Things like bacteria living onto our skin, which we'll get into at a later date. Bacteria provide lots of things for our immune system that are good, and they actually play a large role in our immune system. So we'll talk about those things later. And the last little bit of this is another type of microbial association called a biofilm. Now, biofilms are super neat. Anywhere on the planet that's wet or moist, you can find biofilms there. If you walk across rocks in a river, the slick stuff is diatoms and biofilms and things like that you're walking across. If you don't brush your teeth for a couple of hours and you run your tongue across them and they're slick and slimy, that's bacterial biofilm that's growing on your teeth. They are literally everywhere. But what are they? Now, biofilms are neat. You know good and well that nothing on the surface or on the world is perfectly smooth. When you zoom in down to something, even if it seems perfectly smooth, you zoom in close enough, it's going to be rough, it's going to have some ridges to the surface. And the same thing goes with our teeth. They might look smooth to our eye, but they're not when you zoom in on them. Same with toilet seats and every other surface on the planet. So what's going to happen is there's bacteria everywhere. And as these little bacteria float around the environment on these moist places, eventually one of them is going to stick to that surface. They're going to come across one of those little tiny bumps or grooves or something, and they're going to stick to it. And once one of them sticks, it's the snowball concept. Once one of them sticks, other ones can stick to that one. It's the same way how ice forms. You have to have one ice crystal form, and then all the rest of the ice crystals just stick to that one. If you've ever seen the magic where you crack a bottle of cold water and it instantly turns to ice, that's that same concept. So once one little bacteria sticks, the rest of them all start to blob and grow to stick to it. They start to grow and blob over the environment that they're in. And that allows more bacteria to grow, more bacteria to divide and stick. So once they start growing and sticking well, they will start to divide and grow inside of that biofilm. And once they start growing and dividing inside of that biofilm, the bacteria inside of that biofilm can actually talk to one another. It's a really, really strange thing. But the bacteria over here can communicate with the bacteria over here through all the junk that they're floating around in. So this is all extracellular matrix, dead bacterial cells, all kinds of goodies that bacteria produce. And they can send chemical signals through all that stuff to talk to one another inside of these biofilms. So what will happen is eventually the biofilm will get too large. They will sense that it's too big. Part of the biofilm will break away, float off somewhere else into the environment, and start another biofilm. So very interesting way that these things work. And this is called quorum sensing, talking to one another. So this is a biofilm inside of your toilet. It may seem perfectly smooth, but the little tiny ridges inside of and bumps and things on the porcelain inside of your toilet, the bacteria can stick to. And that's what they're growing on. So this is another biofilm. Biofilms on your teeth. If you don't clean your teeth, what will eventually happen is the bacteria biofilms will die. And as they die, their little bodies, all that junk and stuff, hardens to form plaque. So plaque on your teeth, you can see that up here, is nothing but dead bacterial cells and dead bacterial biofilm that have gathered and hardened around your teeth. Now this stuff can be very, very dangerous to dental health. Your teeth get weak. They depend on this plaque to keep themselves structurally supported. And then when the plaque is removed, they are not standing up very strong anymore. They can fall out and get weak and wiggly and things like that. So keeping your teeth clean is a very important thing, and that's partly why. But very interesting form of biofilms. So that's all I've really got for this lecture for you guys over how bacteria reproduce, how they divide, how we measure that growth, how we can control it and manipulate it in a lab, and then some of the relationships between organisms and microbes in the environment. So other than that, guys, have a nice rest of your day, and let me know if you have any questions.